Bismillahirrahmanirrahim. Assalamu alaikum. I am Dr. Ahmad Nauman from Attack campus of University of Education. And today in this lecture of analytical chemistry, we are going to study about analysis of variance, which is most commonly known as ANOVA. So in this lecture we will have introduction about analysis of variance and its major application, and we will also study how it is actually performed. And besides analysis of variance, there is another very important test which is commonly used while we work with ANOVA. It is known as F test. So in this lecture we will also briefly overview the F test as well. So let's start with our today's lecture. So before we study what ANOVA is, let's discuss why ANOVA is used. So for example, we have 5 analysts which are determining calcium in a sample using a volumetric method. So because there are 5 different analysts which are determining the calcium, so will there be a difference in the results? Or for example, we have 4 different solvent composition and we are using it for the synthesis of some specific chemicals. So will these different solvent composition will affect the actual yield of a chemical synthesis or no? Okay, let's have another question. So if by using some analytical methods, so we have to determine manganese in a given sample, and a person a same analyst is actually going to determine the manganese in a sample. However, he will use 3 different techniques or 3 different instruments or 3 different methods. So whether that use of different methods will give same results or there will be a difference in those methods. And we have one more question. So if we are detecting or measuring the fluorescence of a complex ion, but we are determining it at 6 different pH values, so whether that fluorescence of that complex will show different results at different pH or not. So we have these kind of different sorts of problems in our lab work, and in how we answer that. Actually, these are the situations where we need the use of ANOVA, the analysis of variations. So the above 4 examples are the typical examples of the applications of ANOVA where we use ANOVA. So when we have any of these above situations, so to get the answer we use ANOVA. So in each of these situations, the populations or we have different kind of values or characteristics which we are going to measure. For example, in the first example we have different analysts. In the second example we use different solvents. In third we use different methods, and in the last example of fluorescence we measure at different pH. So all of these methods we are the actually the determining parameters or characteristics, we call them factor. For example, in the case of measuring calcium, the factor is the analyst because there are different analysts. And for example, in the measuring of fluorescent at different pH, so the factor is pH. And when we use pH, so we are having different pH. So when we the factor is analyst, so there are 5 different analysts. So these are known as levels. So different factors which are working on it are known as levels. We call it the different values of the factor, we call them as levels. And the third important parameter is the last one, we call it response, like what we are going to measure by using these different factors and different levels. So we call it a response. In the case of calcium, when we are determining, so amount of calcium is the response. In the second example, the synthesis yield is actually the response, and we are measuring the fluorescence intensity at different pH. So pH is factor, different levels of pH are levels, and the fluorescence intensity is the response. So these are the 3 really important components of the ANOVA. Table actually summarizes these 3 important parameters of ANOVA. As you can see, there are factors like analyst, solvent, analytical method, and pH. So these are the factors. And in analyst 1, 2, 3, 4, 5, or pH 1, 2, 3, 4, 5, 6. So these are the different levels of that factor. And response is like what we are measuring. Like in case first example, we are measuring calcium. In second example, we are determining the synthesis yield. And third, we are measuring concentration. And then in the fourth example, we are measuring the fluorescence intensity. So these are the response. So when we draw actually a graph, we know that there are dependent variables or independent variables. So in this case, when performing an ANOVA, so the factors are the independent actually variables, while the response is the dependent variable. So we will always put factor on the x-axis and response is drawn against the y-axis. So we have a figure to explain that how we visualize ANOVA, and in this case we will take the examples of our first actually factor analyst, in which 5 different analysts actually determine the amount of calcium. So we will look into that example on our next slide. So here is a pictorial diagram which shows the results of the ANOVA for determination of the calcium by 5 different analysts. So the 5 analysts, so they are the factors. So we have put them in the x-axis. As you can see, the x-axis is the analyst 1, 2, 3, 4, 5, and for us here the dependent variable is the response, that is the concentration of calcium in millimoles. So we have plot it on the y-axis. So as you can see, in diagram we have analyst 1, 2, 3, 4, 5, and against each analyst you can see 3 points, which means that each analyst performed the experiment in triplicates, or he analyzed the calcium concentration with using 3 different samples. So the analysis was carried out in triplicate, and from this figure actually you can see that, for example, you see the analyst 1, so he has performed 3 experiments, the analyst 1, and there is a difference between the 3 points. So there is a difference of between the values which were analyzed by the response analyst 1, and same is the case for analyst 2, 3, 4, and 5. We can see that their individual determination have some difference. And when we compare analyst 1 with analyst 2, so we can see that their values are also different from each other, and same is the case with 3, 4, 5. So we have 2 phenomena here कि जो analyst कोई चीज़ मेजर कर रहा है, उसकी जो 3 टेस्ट कर रहा है 3 दफ़ा, तो हर दफ़ा जो है वो रिज़ल्ट जो है वो थोड़ा सा डिफरेंट आ रहा है फॉर ईच एनालिस्ट. एंड जब हम कंपेयर करते हैं एक एनालिस्ट के रिजल्ट को दूसरे एनालिस्ट के साथ, तो उसके अंदर भी थोड़ी सी वेरिएशन आ रही है. सो दैट व्हाट वी कैन गेट फ्रॉम दिस डायग्राम, एंड we will actually this is why ANOVA is used to solve these actually these kind of problems. So we will further explain it in our next slide, that what information we get from ANOVA. So the type of ANOVA we have seen in our last slide. So that kind of pictorial description. So we called it as a single factor or one-way ANOVA because we have only one dependent variable. In this case, that was the result of calcium. And the only factor was the analyst. So this is called this is one on the x-axis and one on the y-axis. So we have just two variables, and we call it one-way ANOVA. But there are certain other complex cases. For example, if we are conducting an experiment and we are synthesizing something, and the rate of chemical reaction is actually determined by two factors. One is pH and one is temperature. So now we have three factors. One is the rate of a chemical reaction, and then we have two actually factors here. One is pH and the temperature. So these both are affecting the rate of chemical reaction. So when we draw ANOVA for that kind of example, so we call it as two-way ANOVA. So but in this lecture we will we are discussing only the one-way ANOVA for your easy understanding because the basic principle of one-way and two-way ANOVA is the same. So as we see in the previous slide, that each analyst in the figure, we have 5 analysts, and each analyst actually performs results in triplicate. So he performs 3 analysis. So these are the so we have 3 factor levels. So we call the factor in ANOVA, we call mostly these factor levels as groups. So analyst 1, analyst 2, analyst 3, analyst 4, analyst 5. So we these are the factor levels. So we often also call them the groups. So now what is the basic principle of ANOVA we are applying here? So the basic principle of ANOVA is to compare the variation between groups and within the group. For example, in this case, the example, the basic what we will do in ANOVA is that we will study how much the difference is between the 3 values which are determined by one analyst and second. So we will, this is the variation within the group. So one analyst has performed 3, the second analyst has 3. So between these 3 variations there is some difference. So we will compare this variation with the variation among 5 different analysts. So this is the basic principle of ANOVA. So in the given example, as we have the factor levels or the analysts, so the comparison is variation between analysis and within analyst variation. Here we can see the pictorial representation of ANOVA. So the result of each analyst which are in triplicate, so they are considered a group. So you can see the triangle with each corresponding to each analyst, and then we take the mean of these values and standard deviation of these values to know how much is the variation between the determination performed by each analyst. And then we compare that. As you can see that we have on the right side that we have 1 2 3 4 5, so 5 circles, so which show the mean value of each analyst, and we also know the variation between the analysis of each analyst. And then we compare these 5 mean values from the different analysis with each other. So by this way, by comparison of the variation between a single analyst variation to the different analysis analysis, so we get the information about the accuracy of their analysis. So what we get from ANOVA, what information we are getting from ANOVA. So basic question was is there any difference in the result of 5 analysts who are determining calcium by a volumetric method? So that was the question. So by ANOVA we actually answering this question, that if analysis is performed by 5 different analyses, so whether there will be a difference in their value of result. I mean, there will be difference, so whether it is a significant or not. So if the variation between the group mean is close to the variation within the groups, between the groups mean between different analysts, and within the group mean the 3 different analyses performed by the same analysis. If that variation is very similar, it means that there is not much difference between the results of 5 analysts, and we can say that the results are reliable. However, if the variation between the group means is very large as compared to variation within the group, so then we can say that there is a significant difference between the analysis performed by different analysts. So this is how the ANOVA actually works, and gave us information about variation between groups and within groups, and tell us answers these questions for us. And actually for ANOVA we have to perform some statistical analysis, and the most commonly used actually tool which is used for ANOVA is the F-test. So this is the basic statistical used for ANOVA. So we will briefly discuss this F-test in our coming slides. So now let's learn some little bit about F-test and how we apply it for the measuring the variance and the difference between the two population, and to decide whether there is a difference or not using the ANOVA. So the F-test is basically designed to indicate whether there is a significant difference between two methods based on their standard deviation, and it is defined in terms of variance of the two methods or the 3 methods, whatever we are comparing. So basically we know that we already studied in our previous lecture of variance and standard deviation, that variance is the square of the standard deviation. So if we are going to compare 2 methods and know whether there are significant difference between their 2 results or not, so we carry out an F-test. So basically F-test is the ratio of the variance of the 2 methods. So F is equal to S1 square, which is the square of standard deviation or the variance, and divided by S2 square. So in F-test we actually calculate the standard deviation of method 1, and then take its square, and then divided the square of the standard deviation of the result from the second method. And in S1 we always put the in nominator we will always put the which have the higher value. So S1 will be always the which have the larger value, and S2 will be the smaller value. So we perform an S-test and we get the result. So what happens next? So इससे हमें क्या मिल रहा है कि information क्या मिल रही है? So basically जब आप 2 methods को compare कर रहे हैं और आप उसमें standard deviation calculate करते हैं, और then F-test जो है आप उनकी जो है वो ratio लेते हैं, then what happens कि we have a standard table है which we will show in the next slide. So that actually give you the F values at 95% confidence level. So we calculate the values using this formula, and we actually then compare these values with the values which are given in the table. So अगर तो जो हम values हम calculate कर रहे हैं, अगर उनकी जो values हैं, if they are higher than the tabulated value at that specific confidence level, and the specific degrees of freedom, then हम कहते हैं कि there is a significant difference between the variance of two methods. But if our our calculated value is lower than the tabulated value, then we say that there is no significant difference between those two methods. And what is a degrees of freedom here? So we will use this term, the degrees of freedom, v1, v2. So basically degree of freedom is defined as n minus 1. N is the number of what you say that response or number of measurement, and degree of freedom is 1 minus number of sample minus 1, or number of the response minus 1. So we will discuss that in detail in our next slides. The table I have mentioned the previous slide, which shows values of at 95% confidence level. So here we see that when there is a v1 and v2, which is the degree of freedom. So we calculate the f from our given data, and then we compare with this table, and if the f calculated is higher than the table value, then we say that the variance being compared are significant different. So there is a significant difference between these two methods. And if our calculated value is lower than what we get from this table, so we can say that they are statistically same and there is no significant difference between the two methods. So let's solve an example so that you have better understanding that how it is actually carried out. For example, we have two different methods. One is the method I'm performing. There is some new method I have tried, and I want to compare it with some standard methods. So when I do the analysis, so I perform like in my method I perform 7 analysis, so which means the n is equal to 7 and the degrees of freedom is equal to 6, and the mean value from my experiment or from my method is 127. So now I want to compare it another standard method, which is Falling-Boom method, and which have and from that method, for example, we carried out 6 analysis and the mean is 128. So mean 1 is 127 and from standard method is 128. However, the degrees of freedom will be different because the sample number is 7 in our method and 6 in other method. So in our method 7 minus 1, the degree of freedom will be 6, and in the other method will be 5. So first we will calculate the f value from these two methods, and then we will compare it with the table. So let's first calculate. So first we have to calculate the standard variation or square of the standard deviation from our method. So after we as you can see from the table, we already actually have practiced how to calculate the variance and standard deviation in our lecture on specifically on this topic. So when we calculate the variance for the first method, so we get the value of 8.3. so here the n minus 1, the degree of freedom is 6. And when we calculate the variance from the standard method, so we get a standard variance value of 4.8. So then we carry out the f test. So f test is the you know that we put the higher value on the numerator and denominator. So when we have the 8.3 value is higher and 4.8 is the smaller, so when we perform the f test, so we get the value of 1.73. so this is the difference between the variance of our method and a standard Falling-Boom method. So f gives us the difference between the variance of these two methods. So to now to actually know whether these methods are significantly different or not, so we will compare our value with the tabulated value. So here for our method we have 7 samples, which means 7 minus 6 degrees of freedom. So v1 as you can see, I have already circled it, the 6, and the 5 is for v2. So these are the degree of freedom. So when we take the central where they meet, so we you can see that value at 6 degree of freedom for v1 and 5 degrees of freedom for v2 is 4.95. So you can see that this we have already encircled that. So the 4.95 is the calculated value, the tabulated value, and the calculated value, as we calculated in the previous slide, was 1.73. So from this we know that the calculated value is less than the tabulated value, so we can conclude that there is no significant difference in the precision of these two methods. And the standard deviation, which are the small difference or the variation, are because of the random errors, and they do not depend on the sample. So the both these methods are we can conclude that both these methods are accurate and there is no significant difference. So this is how actually we use the ANOVA and the F-test for comparing two different methods. So this is how we use the ANOVA technique for comparing different methods. So it is really important for the context of all those situations, that when we want to compare two or more methods or two or more different situations. It can also be, it is not only applied in chemistry, but different branches of science and other fields have been applied. For example, when we have a scenario in which we are using different seed varieties, and when we want to know whether different kind of seed gave different yields or they gave similar yields. So for such scenario we also use ANOVA. Similarly, आपके पास mini cars हैं, for example from Toyota है, from Honda है, from Suzuki है, or from any other brand, for example Kia है. So you have 4 cars which have the same actually horsepower, and आप देखना चाह रहे हैं कि उनकी जो mileage है for gasoline, how much different it is, वो similar है या different है. So you are comparing between the automobiles for different companies. So in such scenario also we actually can use ANOVA to know whether there is a significant difference between their mileage or not. So there are these kind of situation where we mostly use ANOVA. For today's lecture, thank you so much for listening and your patience. And these are the references I have used for preparing this lecture. And besides these two books, I have also consulted and used materials from some internet sources as well. So I really hope that you have understand something about ANOVA today. But in case you have some queries and some confusion, so please let me know, and I will inshallah try to answer you and solve those issues for you. Thank you. Allah Hafiz.